actually am a little bit. So today I would like to talk about the importance of genetic quality control in oocytes and show you that it has good, bad, and ugly side. And I think it's very fitting that I will speak about oocytes or eggs at Women's University. What do I mean by gamete quality and how this relates to disease? So actually each gamete has to contain exactly half of the chromosome number, so half of the genome, because when those two gametes meet, they will create a new diploid organism. And actually all the genome aberrations that are acquired by egg and sperm during their development can be transmitted to the embryo, new organism, and can cause various health problems later in the life. And we know probably very well that chromosomal aneuploidies are associated with pregnancy loss. And actually majority of chromosomes, so aneuploidies of majority of chromosomes are actually not compatible with life. Only a handful of chromosomes can produce viable embryos. Most common aneuploidies that also cause developmental disabilities like mental retardation and a lot of other health problems are Down syndrome, which is trisomy of chromosome 21, Edwards syndrome, Patau, Klinefelter, and the only monosomy, which is monosomy of sex chromosomes, Turner syndrome. Also, a lot of de novo mutations in the germline are associated with diseases. They can cause or predispose to various diseases, and that includes cancer. Poor quality or inability to produce healthy mature gametes can produce various reproductive disorders, such as premature ovarian failure or infertility. Also, immature gametes that are stored in our gonads, in testes or ovaries, can be exposed to various environmental or therapeutic genotoxic agents. So probably a lot of food that we eat, things that we breathe, or therapeutic, which is actually mostly case of cancer therapies. Chemotherapy and radiation therapy attack DNA, or most of them attack DNA, and they cause germ cell depletion, which again causes ovarian failure in female cancer survivors and as well in males. When we think about the very sperm and egg that created us, we hope that they were perfect. There was no aberrations, so we don't have to worry about diseases. For us women, we really want to produce good eggs, so our babies are healthy. So I think it's quite important to understand how germ cells ensure that they carry the correct and intact genetic information. And those haploid gametes are produced by the very specialized cell division called meiosis, and there are sex differences. Males produce sperm continuously from the spermatogonial stem cell pool throughout their reproductive life, and they produce millions, trillions, I don't even know what's the number, but a lot of sperm. So they go for the quantity. Females produce very limited number of gametes, and they are born with a finite number, so we are only losing our oocytes throughout life. So females actually care about the quality of gametes, because not only we bring half of the genome, we also bring a lot of proteins, RNA, and all the goodies to turn oocyte into a totipotent cell. Those haploid gametes are created, as I say, by the process of meiosis, during which one diploid cell undergoes one round of DNA replication followed by 2 rounds of cell divisions. And this produces 4 haploid cells. And again, there's a difference between males and females. From 1 diploid precursors, there are 4 sperms produced, while in the female, from 1 diploid precursor, there will be only 1 egg produced. And today, I'm going to focus only on the female side, because also females are known to contribute majority of aneuploidies to embryos. In order to segregate homologous chromosomes properly during first meiotic division, homologous chromosomes have to be connected by chiasmata. Chiasmata ensure proper bipolar orientation of homologous chromosomes on the meiotic spindle and subsequent chromosome segregation to the opposite poles. Chiasmata are a physical manifestation of reciprocal DNA exchange between maternal and paternal chromosomes, which are marked here in red and blue. And they are they're also known as crossovers. And crossovers are formed, are generated by meiotic recombination during extended prophase 1. So meiotic prophase 1 actually takes about 4 to 5 days, so it's much longer than the mitotic prophase, and can be divided into multiple substages. Early during leptonema, replicated chromosomes start to condense. They find each other in zygonema. They start to synapse. They form what we call synaptonema complex, something like a zipper that keeps two homologous chromosomes together. Crossovers are formed in pachinema, and in diplonema, once synaptonema complex disassembles, those two homologous chromosomes are kept together like chiasmata. What's missing from this picture, and probably only Laura knows, is the meiotic double-strand breaks. So what makes germ cells very special, in my opinion, is that they can deal with a lot of DNA damage. There is about 300 double-strand breaks generated early during prophase 1, which hopefully you can see all those green dots that are marked here with, in green with RAD51 are sites of DNA damage. Another marker that we use to mark DNA damage is histone H2AX, the phosphorylated version of histone H2AX, which marks chromatin around the double-strand break site. Those breaks are actually necessary. It is a DNA damage, it's very dangerous to cells, but germ cells need that in order to find homologous chromosomes. And those double-strand breaks are also precursors to crossovers. As germ cells progress through prophase 1, all those double-strand breaks are being progressively repaired. And as you can see, the numbers of breaks are diminishing, so that by the end of pachinema, there is no more DNA damage left in germ cells or in oocytes. From those 300 double-strand breaks that are introduced early during meiosis, 23 to 30 are resolved as crossovers, so the end point of meiotic prophase 1. Recombination not only produces crossovers to ensure proper chromosome segregation, it also produces new haplotypes, new combination of alleles, so it's very important for genetic diversity. So there's a lot of DNA damage early, and this damage has to be repaired. What happens if cells are unable to repair DNA damage? Usually failure to repair those double-strand breaks means there will be no crossovers. If there are no crossovers, that usually would mean that the oocyte will be aneuploid. An aneuploid oocyte has a high chance to produce aneuploid embryos, so we don't really want that. So there is so a mechanism evolved that eliminates all those bad eggs before they have a chance to produce embryos. And here I'm showing you oocyte spreads, again from wild-type mouse and 2 mutant mice that we use in the lab. And again, Pakitin oocyte does not have any marks of DNA damage. In case of this mutant, you can see there's still a lot of DNA damage left, and all those chromosomes are sort of not well aligned, so they are not synapsed. In the lab, we have another mutant, which is very unique, because here all the chromosomes can pair. There are even crossovers formed, which I'm not showing you here, but what you can still see is that there's a lot of DNA damage still left on those chromosomes. And you can see those green flares, and probably each of these flares represents one double-strand break site, which is still not repaired. What this means for the female. This is a cross-section through mouse ovary, where you can see developing eggs. In the DMC1 mutant, all this DNA damage causes oocyte depletion. And females of this mutant, they're infertile because they don't have any oocytes left in their ovaries. And the same is for TRP13. Same age animals, they are almost always depleted of oocytes. There are rare survivors that are left, but by the age of puberty, those ovaries are totally depleted of oocytes. So we know that those meiotic checkpoints are there to eliminate bad eggs, so we wanted to know the mechanism and regulation of this quality checkpoint. So we went for an old genetic trick. We wanted to know interaction between two genes to deduct what is the pathway that detects abnormal oocytes and eliminates them. We used our TRP13 mutant as a model because it's a good model for to separate DNA damage from chromosome synapses. So what we did, in the wild type, all oocytes progress through prophase 1 without major problems. They arrest at the end of prophase 1 and form primordial follicles. And those primordial follicles establish all ovarian reserve for the whole reproductive life of the female. The rare abnormal oocytes that do happen during normal wild type, during meiosis in wild type females, they activate meiotic checkpoint and they are eliminated. So those abnormal oocytes do not contribute to the primordial follicle pool. So they are eliminated before they have a chance to grow and produce embryo. In meiotic mutants, such as DMC1 or TRP13, all the oocytes are abnormal. They activate meiotic checkpoint, they are eliminated, and ovaries are empty, as you could see before. Now we have hypothesized if we additionally delete a predicted checkpoint gene, we will be able to disable meiotic checkpoint, prevent oocyte death, and see rescued oocytes in ovaries. And we had a short list of candidate genes, and to make long story short, we found that CHEK2 kinase, which is very well-known kinase, is one of the meiotic DNA checkpoint proteins. CHEK2 is not required for normal development of oocytes. CHEK2 females have normal ovaries, they produce normal offspring. But if we delete CHEK2 in the TRP13 mutant, now we can see we can rescue oocytes. Compared to single TRP13 mutants, we see plenty of oocytes in the ovary. We can also see, maybe you can see those primordial follicles that are present. So we were able to rescue a good number of oocytes, maybe not all of them, but about 1/3 to half of the primordial follicle pool. This is at 3 weeks of age. This is a permanent rescue, because when we look at 2-month-old mouse, that's when we start breeding them, there are still plenty of oocytes in those ovaries. And again, I'm showing you primordial follicles here in the insect. Not only we are rescuing and allowing those oocytes to survive, those oocytes actually produce pups. And when I had my very first litter, I called them miracle babies, because if you consider that TRP13 mutant females are not fertile, those are a miracle. It turns out those females can have multiple litters. You can see here in red, the double knockout females produce multiple consecutive litters. Litter sizes are slightly smaller than our controls. This is mostly because less oocytes are being ovulated, and there's a slight increase in embryo loss. And by the time we had 100 pups, they asked me to stop calling them miracle babies, because it stopped being a miracle. But actually it's not, because those oocytes still have a lot of DNA damage. So we are not changing the fact that those oocytes are damaged. In our double knockout mutants, we still see almost meiotically arrested oocytes with marks for DNA damage. And what's interesting is that in 2-month-old ovaries, from these oocytes we have later primordial follicles, we don't see any marks of DNA damage anymore. So somewhere between where the female was born and the 2 months of age, this DNA damage was repaired. What it means is that the DNA damage, which is lethal to oocytes in the presence of checkpoint, can be repaired when the checkpoint is disabled. So this is a little summary before I move on. It's just to sort of a take-home message that the CHEK2 protein actually protects the integrity of the genome in developing oocytes prior to meiotic arrest. It does not allow oocytes that haven't repaired DNA damage to progress to the meiotic arrest. So initially, when I showed you the meiosis, I didn't show you the female meiosis site. In females, actually works a little bit differently. As oocytes progress through prophase 1, at the end of prophase 1, they arrest and dictate, and then resume meiosis after puberty. So we have shown that CHEK2 acts during prophase 1, but we don't know what happens later. What we do know is that dictate arrested oocytes in primordial follicles are very sensitive to induced DNA damage, like the damage induced by radiation. So we wanted to know if CHEK2 also plays a role in meiotically arrested oocytes. So we have hypothesized that those primordial follicles are so sensitive to DNA damage because they still have the same checkpoint operating as during prophase 1, which usually deals with hundreds of double-strand breaks. So what we did, we used radiation to induce DNA damage in CHEK2 deficient mice. First, I will show you what happens in wild-type mouse ovaries. This is a cross-section through a wild-type female ovary, a control one, where you have marked primordial follicles, and the oocyte sits in the middle. After radiation, and we use 0.45 Grays, 1 week after radiation, all those follicles are depleted of oocytes. So all those oocytes died within 1 week after radiation. This is sort of another way of showing that. We have marked oocytes with P63 and MVH, which are markers for oocytes. And again, you can see that 1 week after radiation, all those small cells, which are the primordial follicles, are gone. So, so they are very sensitive to radiation. And actually 0.2 Grays of radiation can already eliminate oocytes. So what happens in CHEK2 deficient mice? All those oocytes survive. What you can see here, those ovaries, they don't look any different from the control ones. All the follicles contain oocytes. And again, you can see that here. And not only, again, those oocytes survive, they can produce viable pups. And we haven't seen any developmental changes in those mice. The litter sizes of those irradiated females were not different than the non-irradiated controls. So this all looks good and tells us that CHEK2 deficient oocytes can survive radiation and that those oocytes are good and they can produce pups. So this made us think that maybe CHEK2 inhibition can be used as a fertoprotective therapy. Right now, there are not a lot of methods that actually preserve oocyte in vivo. Most of therapies focus on embryo freezing, oocyte freezing, or cryopreservation of ovarian slices. So I think it has a promising, it's very promising therapy. So what we decided to do, actually test different CHEK2 inhibitors. There's a lot of inhibitors available on the market, so we selected a few of them and tried sort of first in an ex vivo system. Here I'm showing you sections from in vitro cultured ovaries that we cultured in the presence of DMSO, which is a vehicle for our inhibitors. And this is a section from the non-irradiated ovary. And you can see again, staying with MVH and P63 primordial follicles, and the oocyte is in green. When we use one of our CHEK2 inhibitors at a lower concentration, those oocytes still die. You can probably still see those empty follicles. But when we use the same inhibitor at a slightly higher dose, now you can see some oocytes remaining in ovaries. So all those primordial follicles marked here, those are the primordial follicles. So this is very promising and tells us that maybe we can use CHEK2 inhibition to protect oocytes in the ovary. So at the beginning, I mentioned that this genetic quality checkpoint in oocytes has the good, the bad, and the ugly side, not to be mistaken with the Italian Western movie. So the good side of the checkpoint is that it does ensure genetic quality of gametes, which we really need. Potentially, a mutation in the checkpoint gene can cause females having bad eggs. So if there is a recurrent problems with pregnancy loss, maybe it's a problem with the checkpoint, that those bad eggs are allowed to develop. The bad side is that it seems that this checkpoint can sometimes be overprotective, like in the case of TRP13. The chromosomes are synapsed, crossovers are formed, but the DNA damage kills the oocyte. And when it turns out that this DNA damage can be repaired. And the ugly side is that it eliminates oocytes which are exposed to exogenous DNA damage. And this can be radiation, this can be chemotherapy, maybe it will be space travel, or maybe nuclear reactor breakdowns. There's a lot of radiation that oocytes can take, and maybe having something that will protect them in vivo will be a good therapy. And finally, I'd like to acknowledge my colleagues at Cornell University, Vera Rinaldi and John Schimenti, and my new very tiny lab at Jackson Laboratory. I started in January, so we are still very small, that worked with me. And Korea for giving me a really nice vacation. So this is my favorite place. It was really beautiful. Thank you. I can take your questions.
Thank you very much. And